on their two X chromosomes. So now we'll look at the pedigree. Okay, let's remember that in a pedigree, one thing you're looking at is this. If you got a parent that's got, or two parents that both have the same trait, this time I'll make it the colored in trait, and any one of their kids has the colored in trait, this colored in trait must be recessive. Okay, now if we've established that and we're trying to figure out whether it's sex linked or not, now we look at patterns between parents and kids where we're looking at their gender. Okay, so here, this pattern established that colored in symbols mean they have the recessive phenotype. That means dad's got the dominant phenotype. If this was a sex-linked trait and dad had a dominant gene for his one and only gene for this trait, he could not have a daughter that has the recessive trait, because to be a daughter, she has to get two X chromosomes, and this couldn't happen. So this pattern would tell you it's not sex-linked. There are no patterns that tell you that it's for sure sex-linked. One of the patterns that, after you've established that the colored in is recessive, one of the patterns that says this might be sex-linked is if the only people that have it are males. And they don't all have it, but some of the males have it. I mean, only males have it. That would make it real obvious, right? That would also mean this mom is a carrier, right? She's got, two different genes here, and she gave that one to this son, and that's why he had the recessive trait. So there are patterns that can tell you sex-linked, but there are clear patterns that can tell you it's not sex-linked, right? And here's the other one. If you've established that the colored in is recessive, you establish that from some other pattern like this. Some of the parents in the pedigree were like this. So mom is recessive, right? Dad is, let's say, dominant, right? Mom cannot have a son with the dominant gene. If it's sex-linked, the X chromosome that this son got came from mom, and if she's recessive, that means both of her genes had to be recessive, and all of her sons would get that recessive gene, and that would be their only gene. So this pattern between moms and sons is the same as this pattern between dads and daughters. That tells you this can't be sex-linked, it has to be autosomal. Okay, so a chi-square thing. I'm going to do this now, and this might cause me to split this into two different little PowerPoint note things. So here's the thing that is most likely, since they've taken choice chambers out of the equation, I think, because choice chamber stuff is an ecology kind of question, and they took that unit out. This is the most likely possibility. If they, ask you for that hated chi-square question, here's the idea, right? Okay. What you have here is an expected set of numbers based on, most likely, one of Mendel's laws, or maybe even both of Mendel's laws. Okay. Here we're only talking about one trait, right? So this is not the law of independent assortment. We're only talking about one trait, one pair of genes. So this is law of segregation. What do you expect if you get this or that, right? Well, here they're telling you this is sex-linked, that red is dominant, that mom is red-eyed, and she's a carrier, if you will, of this trait, right? And dad is red-eyed, and that's all they have to tell you, because whatever his phenotype is, his one and only gene is that, okay? So now, what do you expect if you cross these? Well, you expect, right? This is what you figure out. First, see what you're given. Those numbers are on the last page. They'll come up again. You have to figure out what you expect based on the laws of heredity, right? And then you plug it into that equation. So we had this. These were what we got. These are what we observed, right? You had 200 total flies, and that's what you observed. So if your prediction is based on this Punnett square from these two parents that we just described, then you would expect 1, 2, 3 red-eyed for every 1 white-eyed. 3 to 1, 3/4 to 1/4. And out of 200 total, this is 3/4 and this is 1/4. So these are your expected numbers. These are your observed numbers. When you plug them into this formula, if you do the math right, you get these numbers for that calculation, these numbers for that calculation, this total number, and then you go to this table, if you remember, and look how your number compares to some number in this table. Which number? Well, that depends on two things. Again, a p-value, which if you're given that, which I forget if the question said, to be honest, so I'm going to pick 0.05, and then your correct calculation of the degrees of freedom, which is what this row is, and that is always based on, again, how many outcomes are there? 2 in this case. Degrees of freedom equals the number of outcomes minus 1. So we're at 1 degree of freedom. 0.05384 is our chi-square number, which we're going to compare to our calculated chi-square number and see that this is bigger, right? What does bigger mean? These two were different, right? Because our chi-square number at its heart and soul is what did we observe and how different was it from what we expected, right? So if it was different, really different, how different? More different than this number would set the bar at, then we come to the conclusion that we didn't get what we expected. Our little results here do not match our hypothesis, so to speak. Support our hypothesis is obviously better. So we must reject our null hypothesis. This would be the null hypothesis in this case. The null hypothesis would be here the one that you predict based on Mendel's laws. Okay, so just a couple other things. They throw in this. Certain human genetic disorders can be attributed to the inheritance of a single affected or mutated allele. And they give you an illustrated example list. Sickle cell, codominance there, right? Tay-Sachs is a simple recessive disease. Huntington's is caused by a dominant gene, right? And the sex-linked one, all of these are just caused by a single gene. But then this one is caused by an entire chromosome's worth of genes being, in this case, extra. And that phenomenon is the result of something that they put in there. They didn't even ask you, do you know what nondisjunction is? Just can you connect it to this? But here's again nondisjunction. The word means non-separation, basically, right? Non-separation can occur in two places in the formation of gametes we know as meiosis. It can occur in meiosis 1, where homologous pairs fail to separate. This cell got both of those big chromosomes. Or it can happen in meiosis 2, where this double-stranded chromosome failed to separate, and this got both of the separated chromatids, and therefore these gametes had an extra chromosome, a missing one, extra, missing. This is what nondisjunction is about, right? If one of these gametes takes part in fertilization, you will then produce an organism with an extra chromosome, or maybe 2, 3, or 4, or they're missing one chromosome, or 2, 3, or 4. It's rare that these organisms can survive having extra or missing chromosomes, but the case that made their list, right, is when 3 of the small number 21 chromosomes, you can see in this, whatever that skull thing there, I forget at the moment, Down syndrome, they name it here, but again, this is not in a blue box. They don't need you to know this memorization-wise. They might tell you this story and ask you, I don't know what they would ask you, to be honest. I doubt you're going to get this question, but there it is. So last time, they say some traits result from genes that aren't in the nucleus. And those genes would be in chloroplasts and mitochondria. And here is a big difference and reason why they don't follow simple Mendelian rules, because this mitochondria only has one chromosome. So we don't have pairs separating. We don't have any of that. And this mitochondria that's going to end up in baby is only coming from egg cell. So those are the two big differences about simple Mendelian rules. These genes on this circular mitochondrial chromosome, or if you're a plant, the chloroplast chromosomes, they only come from mom. And you're only going to get one of them. You're not going to get one from dad at all, right? And mom doesn't have a pair that's going to separate, so it's quite different from Mendelian rules. And again, they say here, even though nothing else in the syllabus about plants do they expect you to know, right? They throw a couple plant parts in here, and just because they said it in there, they won't ask you a definition thing. But if they tell you this story, now they expect you to know that the mitochondria and chloroplast that little baby plant got came from mom. In the ovule is where meiosis happened to form this little gamete that had mitochondria and chloroplast in it, not in dad's sperm cell in the pollen grain. And so again, mitochondria determined traits and chloroplast determined traits are only maternally inherited. And if this is a pedigree kind of thing, and mom has this trait, right, all of her kids will have it. Any mom in this pedigree that has this trait, all of their kids will have it, males or females. That's the sign of um, this kind of non-nuclear inheritance. I doubt they'll ask you, but it's in the blue box, so there it is. Whew. Well, we haven't even got to this stuff yet, but this is, it's kind of fits here. You've probably heard enough about this, but we got one unit to go, and so keep flossing, keep towing the line, keep touching the line, and I'll catch you next week. I got one practice video to make, and then I'm done for this week. Bye.